Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. This is the cell cycle lecture series. Based on the comments from the previous lecture, in this lecture we will focus on cell cycle on the concerned topics as well as we will touch some other important points. Eukaryotic cell cycle is regulated by periodic synthesis and destruction of the cycles. So the cyclin, the heart of the cell cycle that controls or regulates the cell cycle. The name, the terminology is self-explanatory, that its concentration changes during the cell cycle. We have the various types of cell cyclins like the cell cyclin A, B, D, E, and in the various phases of the cell cycle, their concentration increases, then decreases based on the synthesis and the degradation. It is a very unique 100 amino acid conjugate sequence that is called the cyclin box. And I discussed in my previous lecture that the mitogen that controls the cell cycle in the early G1 phase. What are mitogen? These are the growth promoting factors. Like in the early G1 phase, the growth factor, it can be external as well as the internal growth factor through the mitogen activated protein kinase, the MAP kinase. The sequence of pathways, the RAS, RAF, MAP, or they lead to the cyclin D synthesis. And when the concentration of the cyclin D increases, it binds to the cyclin dependent kinase 4 and 6 and their activity increases. And this enhances, facilitates, or this led the cell cycle to continue. So, like this mitogens, there are anti-mitogens, like the TGF-beta. TGF-beta activates the protein 27, which inhibit the late J1 food. So, TGF-beta is an anti-mitogen. Next, we will discuss what will happen if we withdraw the mitogen in the early G1 phase. When we withdraw the mitogen in the early G1 phase, there is the accumulation of the protein 15 and the protein 16. And we know these are the inhibitor of the cell cycles or these are the tumor suppressor genes. So when their concentration accumulates, they inhibit the cyclin D dependent activity of the cyclin dependent kinase 4 and 6. Thus, there is an inhibition in the proceedings of the G1 phase. When the G1 phase proceedings inhibited, the cell can go into the G1 arrest or it is called the G0 phase. It does not proceed or it does not cross the restriction point to continue the cell cycle. So there are a lot of cells, there are a lot of type of cells like the terminally differentiated cells and other cells which are found in the G1 arrest or the G0 phase. So some of the cells, one example is the fibroblast, which is arrest at the G1 or at the G0 phase. But when there is a wound, the wound leads to the secretion of a mitogen or a growth promoting factor, the platelet-derived growth factor. And when this factor is available, a mitogen is available, so fibroblasts re-enter the cell cycle and it lets the cell cycle to move further so that there is a proliferation of cells and there is a wound healing takes place. Then we have discussed the two important checkpoints. What is the role of the checkpoint? This is to prevent to pass the damaged DNA into the next cell. So it allows the cell to repair the DNA. So if the DNA cannot be repaired, the cell is programmed to go into the programmed cell death or the apoptosis. So before we have discussed two important checkpoints, the G1S and the G2M. And when you will study the DNA replication and DNA repair, you will note that DNA replication is not completed till all the errors because of the DNA replication process are not yet rectified. So this can also be considered as a checkpoint. And another checkpoint is found in the during the mitosis cell division. So before going into, we should know that where the DNA damage checkpoints are important because damaged DNA is not replicated and pass into the daughter cells. So the objective of the cell cycle is not to let the damaged DNA to be transferred into the daughter cells. Then coming to the spindle assembly checkpoint that is found the end of the mitosis. So in this checkpoint, what is the objective of this checkpoint? Alignment of chromosomes on the mitotic spindle, ensuring complete set of chromosomes is distributed accurately to the daughter cells. And the failure of one or more chromosomes to align properly on the spindle, the mitosis to arrest at metaphase stage prior to segregation. So the objective of the spindle assembly checkpoint is not let unequal distribution of chromosomes or not the accurate distribution of the chromosomes into the daughter cells. So the cell arrests itself at the late or the end of the mitosis stage. It is known as the spindle assembly checkpoints. Next, one of the questions of the previous lecture is that there is a G2 arrest. So in the vertebrate oocyte, this G2 arrest is found that lasts for several decades in the human. So as we discussed in case of the mitogen absence, G1 arrest. So if there are not all the factors present at the G2 phase, there are some cells that do not proceed to the mitosis phase. So it happens one of the special case the vertebrate oocyte, it needs a special hormone so that all the requirements to proceed into the next phase is completed. So as long as the individual does not attain a specific age, the hormone is not secreted. So the cell is found, the oocyte is found in the G2 phase and it is arrested. And once that hormone is secreted, the cell enters the cell cycle and it leads into the mitosis and the generation of the second polar body takes place. Then, what is the purpose of the cell cycle? The purpose of the cell cycle is the replication of the DNA or the DNA duplication. So it is a very important stage and we have discussed 3 out of the 4 means checkpoints are dedicated to the complete repair of the DNA. Even the spindle assembly checkpoints is purpose is to distribute the DNA in the equal and the accurate proportion into the two daughter cells. So DNA replication is very important. So the synthesis phase or the DNA replication initiates when the SCF ubiquitin protein ligase is activated that leads to the ubiquitination of the S phase CTG inhibitor. So that if the inhibitors are absent, then it will continue. And in there should be only one replication and only once per cell cycle. So it should not replicate more than once. And we know that the eukaryotic DNA has a multiple origin in the same chromosome. So the initiation at the origin occurs throughout the S phase. The mechanism of the initiation is conserved. We need a origin recognition, then assembly of the pre-replication complex formation. It initiates in the early G1 phase, and the helicase activation, the replisome loading, it happens in the S phase. It means the synthesis of the DNA, it initiates in the beginning of the G1 phase itself, then its spillover or the main part happens in the S phase. So the synthesis of the DNA, it spreads from the early G1 to the S phase. And the checkpoint regulation is to maintain the high fidelity by stabilizing the replication for preventing cell cycle progression during replication stress or the damage. And the initiation, recruitment is called the initiation and subsequent stages of the replication is called the elongation. And coming to the mechanism, as I discussed in the previous slide, that the replication, the synthesis of DNA initiates in the early G1 phase. So we can see this is the cell division has completed and this is the early G1 phase. This is the origin place. So this is a 6-protein origin replication complex that recognizes the origin found in the eukaryotic DNA. And once this is recognized and the ORC binds to the place of the origin, the CDC6 and the CDT21 recruit a 6 or the hexameric mini chromosome maintenance MCM, which is a helicase, to this origin replication complex site. This process is called licensing, by which the pre-replication complex components are assembled on the origin site. So it is a license process. It happens in the RB geometry. One of the inhibitor of this process is the geminin. Then the late G1, the CDK, cyclin dependent kinase, and the DDK, DB4 dependent kinase. They get activated and they load the DNA polymerase complex at the site of the origin. So in this process, the CDK, it activates the DNA polymerase and the SID 2.3, and the DDK, it activates the helicase. Because of this, the A domain of the helicase is pushed out and the replisome is formed. Replisome is the replication complex that polymerizes the DNA. So in the late G1 phase, there is a helicase activity is happening or the helicase is activated. The another function of the CDK is it inhibits the further licensing. Further licensing means it inhibits the assembly process at the origin of replication till it is not completed. So there are two functions. And this process of the replication, it is the rate of the replication. It is adjusted at the initiation site. Means here, the rate of replication is initiated here. Like during the embryo or the embryo development, the rate of the replication is twice, at least twice than that of the adult. Thank you.